Thank you, Joanne, and it's a pleasure to be here and to participate in this meeting. It's great to have it here in Baltimore, so I'm sure someone has already said it, but welcome to Baltimore. So I'm going to cover a good bit of ground in the next few minutes, and I will say that my understanding is that the audience is quite heterogeneous in terms of its expertise. So I'm going to have a little bit of Biology 101 at the beginning, just to make sure that you all have a chance to understand some of the stuff that we will talk about as I go forward. So I'm going to talk, I've changed the title a little bit, and increasingly genetics is at the lead of what I would call sequence-based biology, that is using both the genome sequence and the RNA sequence to understand the role of genes in biology and the consequences of genetic variation on human phenotypes. So it's really a whole new area of biology and medicine, and we are privileged to be standing at the front door, and we have an exciting future in front of us in terms of what we, I think, will learn as we go forward. That's not to say there are not pitfalls along the way, so we have to try to be as smart as we can to avoid them. So I put in these disclosures. The only thing I will disclose is that I'm enthusiastic about genetics. So now the basic biology. And I think everybody here knows, but just let me make sure that here's a typical cell. Our body is made up of maybe 100 trillion cells. And virtually all cells have a nucleus, and inside that nucleus is the DNA. It's about 1.5 meters of DNA wound up in a fashion that you can utilize. It's always amazing to me. I quit being a fisherman because I spent too much of my time untangling the line, and I don't know how Mother Nature keeps this straight. But the DNA, of course, is on the chromosomes, and we visualize those chromosomes and line them up in a karyotype that looks like this, 46XY. The basic chemistry of DNA is shown here. As you all know, it's made up of 4 bases. GATC typically is what we refer them to. And there's a set of rules that say that it's a double-stranded molecule, and A always is opposite of T or pairs with T, and G always pairs with C, and so forth and so on. So if you have a single strand, if you had broke this double-stranded molecule and split it down the middle, and you put both halves in solution, they would find each other by this complementary pairing of one another. It's really quite powerful, and it is the basis for almost every tool that we use in current molecular biology and genetics. And of course, it's wound up into a double helix as shown on the right. Now, the DNA encodes about 20,000 protein-coding genes, and the proteins are made up of about 20 protein amino acids, and they look sort of like beads on a string, as shown here, starting from the left-hand side, ala, gly, glu, and so forth. And the sequence, the primary sequence of the amino acids in a protein determines how the protein folds, and each protein folds in a very complicated but very prescribed way. And it's necessary for the proteins to fold to to get into sort of functional readiness. If they don't fold properly, they're not functionally active. Now, the central dogma of information transfer in biology is shown here. The order of the amino acids in the proteins is determined by 3 base codons in the DNA. So on the top, you see the DNA, and I've underlined in different colors the 3 base codons. And each of those codons specifies a particular amino acid. The DNA is in the nucleus, so it's copied or transcribed into RNA. RNA goes out into the cytoplasm of the cell, and there those 3 base codons tell the protein synthetic apparatus how to put the protein together in the right order. So if it's a beta globin gene, the RNA then says make beta globin, and that's what you get. So that's the transfer of information from the genome through the RNA into the proteins, and the proteins do the business of the cell. If we have a mutation in DNA, it makes an abnormal RNA and an abnormal protein, and that protein may not function, and then you have a phenotype. Now, the arrangement of genes in the genome is shown here. So in the top line, I have a structural gene. I don't know if this, yeah, there you go. So, and these 4 red blocks are exons. They encode the part of DNA that has the information for making the proteins. So we sometimes refer to them as the protein coding portion of the DNA. And upstream of this 4 exon gene, I have a series of, I have the red and purple regulatory elements that I will come back to periodically. E indicates an enhancer, and P indicates the promoter for this particular gene. Now, when the gene is transcribed into RNA, transcription starts at the first exon and goes the full length of the gene. And then the middle parts, these horizontal lines, which are called introns, are spliced out, and so all of the exons are brought together in a much more compact molecule, and that molecule leaves the nucleus and goes out to the cytoplasm, where it's referred to as a mature mRNA, and that's what has the directions for making proteins. So I'll come back to this, but when you sequence a whole exome, which I'll talk about in a few minutes, you sequence these red boxes, and you don't sequence the introns, nor these regulatory elements, nor the intergenic space, which is not shown on this slide, space between one gene to the next. So some additional definitions before we leave this. The genome, as I think you all know, one way to think of it is it's a haploid set of 23 chromosomes, the 22 autosomes, and a sex chromosome. And each haploid set has about 3.1 billion base pairs, so I get a haploid set for my mother and a haploid set for my father. So the total genomic sequence in the nucleus of my cells measures about 6.2 billion base pairs, 2 copies, one from mother and one from dad. The exome, as I pointed out, is that part of the genome formed only by the exons, and it's a tiny fraction of the whole genome. It's about 1.5% of the total. Genetics, what we're talking about today, is the study of inherited variation, and genomics is the study of genomes, including whole genome sequences, and we'll talk about that as well. Genomics really informs genetics. Okay, so now this is a personal bias, but I think it's a really important point. We frequently talk about genetic disease as if there's some other kind of disease, but from my viewpoint, all disease has a genetic component. So we can talk about diseases which have a major genetic component, the Mendelian or monogenic diseases. Those are diseases in which variation in a single gene is sufficient to cause the phenotype. In general, they're individually rare, and we know of more than 5,000, and I would submit, and it's time for a different talk, that there are many, many more unrecognized Mendelian phenotypes that we have to learn about. They're transmitted according to Mendelian rules, exactly the same as Mendel worked out in his pea plants in 1865. Really amazing, a nice piece of work, right? Complex traits, at sort of the other end of the spectrum, are the common complex disorders largely of adult life, and they typically, the genetic contribution there is a collection of risk variants, variants that in and of themselves can't cause the phenotype, but in the proper combination of risk variants, together with a set of environmental experiences, can cause the phenotype. So genetics is at work there, it's just harder to see, and it's not so overriding as in the Mendelian disorders. Okay, now another way to think of this is what I would call the gradient of phenotypic effects. So here we have different genes along the x-axis, and we have here the strength of phenotypic effect. That is, how much effect on the phenotype can a variant in a particular gene have? And you'll see there's a gradient. So for some genes, a variant causes a major effect on the phenotype. And those genes, of course, and those variants are the monogenic variants, the Mendelian variants. At the other end of the spectrum, we have genes in which variants have a modest effect, or perhaps in and of themselves no discernible effect on the phenotype. And it's only when they occur in combination with other variants and environmental experiences that we see a phenotype that we typically would call a common complex trait. So you have this, think of a gradient of phenotypic effect, and different variants in different genes are at different places on this phenotypic spectrum. What I'm going to talk about for the next few minutes is largely related to Mendelian disease, because that's the area that we know the most about. but I will try to remember at the end to sort of translate it into the common complex traits. So although we think of Mendelian disease as being rare, it has become increasingly more prominent over the last few years in the standard medical literature. So this I just took about a year ago, actually. I just went through the 4 editions of the New England Journal of Medicine for the month of January. I think this was January 2015, and I just collected in those 4 issues how much Mendelian disease was in those in that journal. And you can see there's actually a lot. And although most of it is childhood illness, there's even one that is the NPC1L1 has to do with mutations that cause coronary artery disease in adults. So although we typically think of Mendelian disease as being largely in the pediatric age range, as we get more sophisticated, we see Mendelian disease in the adult age range. And we even see increasing concern in the medical community about what it means to do genetic testing and to identify these variants, some of which cause a phenotype and others of which provide risk for a phenotype. So there's a lot of Mendelian disease and a lot of interest in Mendelian disease in the general sort of medical literature, and you could ask what is the source of this increasing prominence of Mendelian disease? And I would argue, first of all, the Human Genome Project, which was complete in 2003, provided a reference human sequence, so it made it a lot possible for us to recognize genetic variants that cause phenotypes. And of course, sequencing technology has evolved dramatically over the last 2 decades with a tremendous several orders of magnitude reduction in cost and increase in throughput, that is, the speed at which we can do the sequencing and analyze it. And there have been a number of projects, the 1000 Genome Project, the HapMap Project, which have focused on identifying and enumerating the variation in our species, the appreciation of the extent of, quotes, normal human genetic variation, which is quite a lot of human variation. And then the last item is the development of genomic and genetic strategies to identify responsible variants in genes that cause Mendelian disease. And we'll talk about that as I go forward. So when does one think of, when in the clinic does one think of Mendelian disease? Well, as I said, most of them are early onset. They're egregious, by and large, they are egregious defects in our homeostatic capabilities, so they cause a problem early on in life. Most or many have a positive family history with multiple affected SIBs and or generations. As I was saying to Nada, I put the slide in just because I love this picture of this family, so I like to get it in here. Consanguinity, of course, increases our suspicion, and the phenotype for many Mendelian diseases includes multiple systems that we can't typically connect in an immediately straightforward way. So I'll just give one case because I think it sort of puts it in a clinical perspective. And this is a case that I was involved in, and I find it very educationally laden. So this was a 34-year-old male, a healthy man who worked as a computer software engineer. He had a 10-day history of fever and pharyngitis. He was treated with antibiotics, a couple different rounds of antibiotics, and his personal physician, in desperation, changed his antibiotics and also gave him a big blast of steroids. Following that, he developed a mild confusion, and his doctor had the wisdom to send him to a local ER, and they had the wisdom for measuring his ammonia, which was 280 micromolar, which is about 10 times normal. Okay, and that would certainly make you confused, and it puts you on the way to coma. And he had respiratory alkalosis, which is consistent with ammonia accumulation. They referred him to the Johns Hopkins Hospital MICU, where by the time he arrived 2 hours later, he was already in early coma, and a CT scan on the way in the hospital showed that he already had mild but definite cerebral edema. So basically, for the non-medical folks in the audience, this is a man who had one toe sort of over the casket. And his ammonia had already risen in that interval to 420 micromolar, about 20 times normal. So I saw him with then a genetics fellow, Hans Bjornsson. We walked over and saw him about 20 minutes after he hit the MICU. And so the first thing we wanted to know, being geneticists, is what about the family history? And we talked to a medical student who had worked him up. And we were reassured that the family history was, quotes, negative. So one take home from this lecture is that the only people who have a negative family history are people that are adopted and don't know anything about their biological family. Everybody else has a family history that has information in it. So that's what I told him. And I said, the parents were already, his relatives were already out in the waiting room, and I said, go out there and ask this question and that question. So he came back, and the pedigree was actually a good bit more expansive than originally concerned. And there's our proband with the red arrow, and he actually had 3 male siblings that were all dead. Now the twins died in childbirth, and they were clearly, this was clearly an obstetrical death. It was not a metabolic death, so we'll set those aside. Now the brother died when he was 14 years old, and the medical student said, don't worry, he died of drowning. So I said, what did I say? Well, I said, what do you mean? 14-year-old kids don't typically die of drowning. Go out there and ask him how it was that he drowned at age 14. And to make a long story short, it turned out this boy was on an outward bound experience. He became progressively confused during his outward bound trip. They camped beside a lake, and in the morning his colleagues found him floating. He'd fallen off the dock in the middle of the night and he had drowned. Ultimately, because it was an accidental death, he had a post. And the county coroner in this remote part of West Virginia, I think it was, noted, curiously, that he clearly had died of drowning, but he had cerebral edema, which is something that you don't typically see in someone who drowned. Well, so it turned out that this man had adult onset OTC deficiency. We were able to get a baby tooth of the 14-year-old brother and show that both of them have a mutation in the OTC promoter that impaired just modestly their homeostatic ability, and only when they were really stressed, in the case of our patient by the severe febrile illness that had gone on for some time, and perhaps by the steroid dose on top of it, was their urea cycle unable to maintain homeostasis. So, and of course, his mother is a carrier. It turns out her sister is a carrier, and she has 2 males who are at risk. We don't know what their situation is beyond that. So the bottom line is there's a lot of Mendelian disease out there, and the more we educate our colleagues to look for it and the more tools we have to find it, the more we will find. So how do we find the responsible genes and variants? So there are 2, in my view, 2 breakthrough papers. The first one was the top one here by Sarah Eng from Debbie Nickerson and Jay Shanduri, and they showed that you can use a whole exome sequencing to identify a gene and variants responsible for Mendelian phenotypes. And they did a great experiment. They picked a known phenotype for which the disease gene was known. They sequenced one affected individual, then another affected individual, then another affected individual, and each time they compared the variants in each individual, and they looked for overlaps. And by the time they sequenced 4 unrelated individuals, there was only one gene that had a loss of function variant in it that was shared by all 4 of those unrelated individuals, and that gene was the gene that they already knew beforehand was the responsible gene. So that said, we can use whole exome sequencing and a genomic approach and find the responsible variants. Now, in another paper, actually by the author, by the next speaker, we did whole genome sequencing, and the only thing that's different about this paper is that we incorporated not only genomics, but basic genetic principles in terms of who was at risk and who wasn't at risk to help us interpret the results and led us, again, to the affected individual and the responsible gene. So those 2 papers together, in my view, provide the foundation for a lot of work that's going on right now. So that gets us to the rise, what I call the rise of clinical DNA sequencing, which I'm sure you all know is being used increasingly in clinical medicine. And I organize DNA sequencing by target type. So we can sequence a single disease gene. We make a diagnosis. We think that diagnosis is caused by mutations in this gene, and we order a sequence of that disease gene. Think BRCA1 for breast cancer. We can make a diagnosis, and we know that this phenotype can be caused by mutations in several genes. So let's say cardiomyopathy and maybe 20 or 30 genes that are known to cause cardiomyopathy, and we order a panel in which those 20 or 30 genes are sequenced. The rest of the genome is not sequenced, or at least not reported, and we only look at those 20 genes which are already known to cause cardiomyopathy, and we look for pathological variants in those genes. Or we don't really have a diagnosis, so we do whole exome sequencing the entire exome. Remember, we talked about the exome at the beginning with the splice sites and the flanking each exon, and we expect that about 85% of Mendelian variants, that is, variants that cause Mendelian disease, will be found in the exome. So that's a pretty good place to look. Or we could order whole genome sequencing, which largely up until now has been a research tool, but you get the whole ball of wax with that. You get all the exons, you get the introns, you get the regulatory sequences, remember that enhancer, the promoter, all of the stuff. So a lot more data, about 100 times more data than in the whole exome. So one thing that's come out of this, let me make one other point, that there's a difference in my view between research whole exome sequencing and clinical whole exome sequencing. So in the research whole exome sequencing, we don't know what the disease gene is, we don't really have a clue, so we sequence the entire exome and we analyze it with the notion that the causative variant could be anywhere in that exome. If you just do an exome on any person, you will find about 80,000 variants, 70 to 90,000 variants from the reference sequence. So the trick is to sort through that large number of variants and find the one that's responsible. And typically we use family relationships and a lot of other stuff to figure out what's going on. A clinical whole exome, the type that's offered by a commercial lab, does the same sequencing, but they look only at those genes that are known to be disease causing. So they don't, although they call it a whole exome, and although they sequence the whole exome, the only ones they analyze are those genes which are already tagged as causing Mendelian disease. And as I'll show you in a minute, that's about 3,600 genes. It's really not a whole exome. It's about 15% of the exome. So when you do that, or any of these sequences, there's two uncomfortable realities that have turned up, and many of you know about these. One is that you will find variants that are difficult to interpret. In fact, you don't know how to interpret them, and they're called variants of unknown significance. And it's important to let the patient, the person who's being sequenced, know that you expect to find variants that you will not be able to interpret. And then the second category are what are called incidental findings. These are variants that you can interpret, and they have nothing to do with the reason that you sequence the patient, but they may have significant medical consequences for that patient and that family. So these would be the 56 so-called actionable genes that the American College of Medical Genetics and Genomics defined, and some people have added on additional genes in that collection. So you sequence a child for explaining intellectual disability, and you find a pathological mutation in BRCA1, and so now you know this family is segregating a BRCA1 variant. They said, wait a minute, I'm trying to find out my intellectual disability etiology. Now you're telling me I have to deal with a risk for breast cancer and pancreatic cancer in the family. So if you go back to the sequencing approaches, how does this all sort out? So if you're sequencing a single gene, for example, BRCA1, the cost will be a few hundred to a couple $1,000s. It's less expensive if that's the right gene. You will find fewer variants of unknown significance because you're only looking at that one gene. And you will find no incidental findings because, again, you're looking at a gene that's already tagged by disease. If you use a panel, let's say a cardiomyopathy panel, maybe 25 cardiomyopathy genes, it'll cost you a few hundred to a few $1,000s. It's a broader net. It's less expensive on a per gene basis. You will find more variants of unknown significance, and you, again, will find, however, no incidental findings because you limited your search just to that specific set of diseases. A whole exome sequence, by contrast, will cost you around $5K, $5 to $7K. It's a much broader net. It's a bargain on the per gene basis. You will find for sure many variants of unknown significance, And you have an appreciable chance of finding incidental findings, a few percent, most studies show. And then whole genome sequencing, we have less experience with this in clinical medicine, but it's essentially the broadest net we know how to throw right now. And it will cost somewhere between $5,000 and $15,000. It's as I say, the broadest net we know about. Hard to interpret, many variants of unknown significance, and you're likely to find incidental findings as well. So keep those features in mind as you're picking your tool. Now, what is so clinical exomes are very much in vogue now, and let's look at the outcome. So the initial paper to look carefully at this came from Baylor College of Medicine. They looked at their first 2,000 samples in their clinical lab, and the bottom line here is they made a molecular diagnosis in about 25% of patients. This thing is, see that third bullet down, and interestingly, 58% of those diagnoses depended on disease genes that had just been recognized in the last couple of years. So that gives you an idea of the pace of progress. The inheritance pattern is there. The second interesting thing is that 23 patients, 4.6% of the total, actually were difficult to diagnose because they had two Mendelian disorders. Typically in medicine, we're taught to make the most conservative diagnosis, but they were having problems with these because they had two different diseases. I'm not going to go through the slide, but GeneDx did the same thing with 3,528 probands, published a little bit later, and basically they found the same thing, about a 25% success rate. Baylor went back and did it on adult patients, and they found a little bit lower success rate, 17%, but explained a lot of problems in this adult population. It was more likely to yield a result in younger adults as opposed to older adults. So it's a pretty powerful diagnostic tool. Now then you have to ask yourself, what is the value of a precise diagnosis? It shortcuts the diagnostic workup. I mean, it stops it in its tracks at that place. It ends the uncertainty of the diagnostic odyssey. It provides a biological explanation for the problem. It focuses patient management, and it informs the family of recurrence risk. So I would submit it's a pretty salutary outcome for a test which is often referred to as being expensive, but in the face of things, I would argue, is not that expensive at all. Here's another paper. You can just look it up. It finds the same thing with sick newborns. They found made a diagnosis in 57% of the people studied. You want to be a little bit careful of those numbers because it depends, your success rate depends a lot on how your criteria are for who you sequence. We could get the success rate up to about 90% if we only sequenced people that we were sure had a Mendelian disorder. So the value of a diagnosis, for me, this makes this point. This boy is a patient of mine. I followed him, I continued to follow him for, I think, 35 years. I didn't know what he had, except I knew that his mitochondria weren't working well. We finally were able to get his mitochondrial genome sequenced, and that turned out to be normal. We finally figured out a way to get his whole exome sequenced, and turns out he has a form of mitochondrial depletion syndrome, so-called mitochondrial depletion syndrome type 13. And so after 35 years, I was able to sit in the room with his mother and tell her, this is the source of your boy's problem. She was enormously relieved. We couldn't do anything about it, but for her, the diagnostic odyssey had finally stopped after 35 years. I hope we can do better next time. So one way to do better is to use research approaches to find the disease genes and variants. We're part of this Centers for Mendelian Genomics, which is funded by NHGRI and NHLBI to search in families that we think have Mendelian phenotypes to try to find novel disease genes. Our center is called the Baylor Hopkins Center for Mendelian Genomics, and that's the website there. You're welcome to submit cases, and if we can fit them into our pipeline, we'll do the sequencing and send you back the results for free. The current scorecard is shown here from OMIM, and I just point out that here, as of last night, There were 3,633 disease genes known. That's about 18% of the total. And it explains over 5,000 Mendelian phenotypes. So the way I look at that is that's good progress, but we have a long way to go. And new phenotypes come in at about 300 per year into OMIM. I'm going to skip this slide. If you look at the summary of what the first 4 years of funding did for the Centers for Mendelian Genomics, this is just our center. The bottom line here is what have we discovered, and we found in sequencing over 9,000 samples, we found 468 disease genes, including 222 which were novel. The others were disease genes already known, but these patients had somewhat different phenotypes than were associated with those disease genes, so-called phenotypic expansion. So we're making good progress, but we have a ways to go. So finding a disease gene connects the genes to the phenotypes. It connects the phenotype to a biological system and tells you something about what the system does both normally and when it's perturbed. It unravels locus heterogeneity. It enables precise diagnosis and counseling. It's the first step towards informed treatment. Maybe a long path, but it's the first step. And it's a powerful research stimulus from bench to bedside. Now, some emerging ideas from this progress. The extent and distribution of genetic variation was even larger than we anticipated. The extent of locus heterogeneity is even larger than we anticipated. There are many examples of phenotypic expansion. That is, we think we know all there is to know about a disease because we've studied 3 patients with that disease, and we study another 3 patients, and guess what? We find out some new features of that disease. There's an unexpectedly large role played by copy number variation and de novo mutations. I don't have time to go into that more. But there's a relatively high frequency of 2 diseases occurring in the same difficult-to-diagnose individual. And we've learned a lot about genetic architecture and burden of disease. I can't see that, Jen. I'm done. I'm not quite done, so let me just... So if you want to read about it, there's the reference in the American Journal of Human Genetics a few months ago. Now, I have just, I think, 3 more slides. I want to come back to this diagram of the genome and how it's organized. Remember the difference between the exome and the genome. So we're constantly debating whether we should continue to use whole exome or whole genome. And here's some things to know about whole exome. When you sequence a whole exome, you make what's called a sequencing library. And that involves, remember that complementary capturing of DNA strands, that involves a hybridization step designed to capture all the exonic sequences. And then you wash away everything else. So the other thing to know about whole exome is it's relatively inexpensive, and the interpretation is relatively straightforward. There's a lot of available reference data that we can use to help us interpret the outcome. There are some problems. Not all Mendelian variation is in the exome, so we miss some. The capture is incomplete. It only captures somewhere around 80 to 90% of the exons, so it depends on which reagent you're using for capture. And it is very poor at delineating copy number variation, which is a major source of disease variation. So it's not perfect. Now, what about whole genome sequencing? There's no capture. You just cut up the genome and you sequence all of those pieces. So the aim is to sequence the entire genome, the exons, the regulatory regions, the whole ball of wax. It's more expensive. It's currently 3 to 4 times more expensive than the exome. The evaluation is much more difficult. There's a lot more data, 100-fold more. The rules for interpreting regulatory variants, that is, when you see a sequence variant in an enhancer or promoter, it's very difficult for us right now to interpret it. And the other thing is that there's a real paucity of reference data. So we can go to exact and look at more than 60,000 exomes, but there's no corresponding exact for whole genomes. There will be in a couple weeks, however. So last night, about 4 o'clock in the afternoon, actually, online, this article appeared in PNAS, and it is the first reported sort of exhaustive whole genome sequencing. It's done by Craig Venter. It's a very useful article. He did a proper scientific experiment. He took one individual and sequenced them 200 times and asked how reproducible his data were and all those sorts of things. Here are some takeaway messages. 84% of the genome was sequenced with confidence, including 1 or 2 megabases that are not even listed in the reference genome. 95% of known pathological variants were detected in the individuals he sequenced. Each new whole genome sequence detected 8,579 novel variants, variants that had never been seen before in anybody. That's amazing. They detected in all greater than 150 million variants. Most of them obviously had been observed before, but a good number of them had not. Most rare and or unknown. And they identified regions of the genome that are much less tolerant of variation than others. So presumably those are where selection is working hard, and that's where disease is located. So there's nothing in that article, unless I missed it, about making this available for reference sequencing. I hope that pressure forces him to do that. But I do know that Daniel MacArthur and his team that are responsible for the EXACT database promises to put about 12,000 whole genome sequences in EXACT before ASHG, so that's 2 weeks from now. So I hope I've convinced you that the field is moving fast. You should use this information to help your patients, but you should be aware of the challenges, and you should explain it to the families and the individuals before you do this test. You need a lot of counseling to explain the variation. In my view, whole genome is going to turn out to be the way to go as soon as the cost comes down more and we get maybe a year or two of interpreting under our belt. I leave the interpreting to Nada, so it's easy for me. And these are the people that have participated largely through the Baylor Hopkins Center of Mendelian Genomics. Thank you very much.